Repetitive DNA. Chromosomes, they are highly dynamic. Whole genome can duplicate. The genome of two distinct species, they can merge, they can integrate. An individual can acquire one extra chromosome or more than one extra chromosome. Chromosome can fuse with each other, like the example is human chromosome 2 derived from a fusion of two ancestral primate chromosomes. Chromosomal regions can be inverted or deleted. Segmental and other duplications can also occur in the chromosomes. So chromosomes, they are highly dynamic. Different phenomena usually are seen that the entire genome can replicate or duplicate. There can be addition or deletion of one or more than one chromosomes. There can be inverted repeats. There can be deletion of certain fragments from the chromosomes. All these phenomena are usually seen in case of chromosomes or in case of genomes. There are 5 main classes of the repetitive DNA. They can be in the form of interspersed repeats, which are called as RNA-DNA transposon derived. Approximately 45%, if we take the example of the interspersed repeats, approximately 45% human genome consists of interspersed repeats like lines, signs, and ALU repeats. If we define that what are interspersed repeats, they can be in the form of retrotransposons, which constitute approximately 45% of the human genome and consist of several millions of family members. They play an important role in shaping the structure and evolution of the genomes. Another type of the repetitive DNA can be in the form of processed pseudogenes. These genes have a stop codon or frame shift mutation or loss of promoter activity and do not encode a functional protein. They are also seen in different genomes of the eukaryotes. Pseudogenes, they commonly arise from retrotransposition or following gene duplication and subsequently gene loss. Another type of the repetitive DNA can be in the form of simple sequence repeats. Simple sequence repeats can be microsatellites, it can be mini satellites. If we take the example of microsatellites, there can be repetition of the nucleotide from single nucleotide up to 12 or 13 base pair, while in case of mini satellites, there can be repetition of the DNA sequence from 12-13 nucleotide up to 500 base pair. So these are the examples of microsatellites and mini satellites. There is another type of the repetitive DNA sequence which is called as segmental duplication. Segmental duplications block of about 1 kb up to 300 kb. So these are large blocks of DNA that can vary between 1 kb up to 300 kb, and they are also found in the eukaryotic genes. If we take the example of the segmental duplications, we see that approximately 5% of the human genome consists of segmental duplications. In case of repetitive DNA sequence, we also see blocks of tandem repeats. Blocks of tandem repeats include telomeric and centromeric repeats and can span millions base pair. Often these type of the tandem repeats, they are species specific. These are the repeats which have been seen in the mouse genome, where we can see that in case of mouse genome, there can be highly repetitive DNA. The size of the highly repetitive DNA can be 10 base pair, and it is 10% of the genome. While in case of moderately repeated DNA sequences, the size of the moderately repeated sequence can be between 150 base pair up to 300 base pair, and moderately repeated sequence consists of 20% of the mouse genome. Main classes of the repetitive DNA can be in the form of simple sequence DNA, that can be in the form of DNA transposons. It can be in the form of LTR transposons. It can be non-LTRs like retrotransposons, lines and signs.